Hello and welcome to the lecture 19 of the Learning About Learning lecture series. In the previous lecture we learned about the molecular representation of memory if you had to do that, and what are all the characteristics a given molecule should possess given the fact that there is a molecular turnover, and how can we transpose this problem such that the memory can be encoded at the level of individual molecules, at the same time robust enough to not being getting muddled with the turnover, right, not getting spoiled by the molecular turnover. And we hypothesized that or we actually proposed that such a molecule should have certain characteristics, and then we were looking at alpha chem K2, that is calcium calmodulin kinase 2, and the alpha isoform of that could be a possible candidate for involving in this kind of a function, like encoding, I mean preserving or maintaining that memory in the molecules at the molecular level. Now the question that we raised was that can this alpha chem K2, if you take it out from the mice, can this alpha deletion in the chem K2 show up as a loss in memory? Now that is the question Alcino Silva working with Susumoto Nagawa asked way back in 1992. So they generated the alpha chem K2 knockout mice, and then first thing you want to study is that in doing so you are not affecting the neuronal conduction, right. That is pretty important, because if you had to affect the neuronal conduction, it is much to do with the information, incoming information itself is not coming in. So there is no point of encoding it. So it should not be affecting that, and clearly they show as they as can be seen in this responses, the evoked responses. What we are seeing here are the evoked responses in the knockout versus in the control, and they are classifying they are measuring two different kinds of currents, the NMDA receptor dependent current and non NMDA receptor dependent current. The reason I mean how you would do that is you just block the receptor, the NMDA receptor, and then measure whatever is happening, and that is the background current, and you can subtract that and you can actually generate even the NMDA current characteristics. So these inserts are that NMDA currents in both the normal as well as the mutant. In fact, you can't even make out which is which, that identical they are. So the inputs are all right, there is no problem with the inputs. Now what happens to the LTP, right? The first thing that we want to ask is that do they express LTP, and they asked the same question, and then when you perform this experiment, depending on whichever the measure that you do with the Timon Blish and Lomo study that we have discussed, we talked about measuring one thing, which is the amplitude. You can actually measure the amplitude as a function of the percentage basal line conductance as is done below in these graphs, these guys, or you can measure the rise, the rise of the potential, how fast, the slope so to speak of these potentials, and whatever you measure, what you see is that in the normal animals they are pretty intact, that is exactly what you would expect. However, in the mutant, even though they get this conduction, that is normal, right. So you would get that is there, but they are not able to maintain that higher level, that is where it is, right. It should have been, but it is not there, it is actually down, reduced pretty highly down. And in fact you can go ahead and again measure the NMDA versus the non NMDA currents. What you see is that again that you can do it in the amplitudes. What you see is that the AMPA currents that have the non NMDA currents that have gotten enhanced in these guys, we do not see that there, right, after the induction of the LTP, that is not happening there at all. clearly their ability to maintain the higher conductance is not there, is lost, and the only thing that you have done is you have taken away the gene for making alpha ChemK2 protein in these mice. That is astounding, that is really astounding at when this was done at 1992. For the first time you can actually show that you can knock out a simple molecule, right, a gene for a protection of a molecule, and these organisms becomes like HM. We are not removing the hippocampus, we are not lesioning out any brain region, we removed a gene and the resultant that we have is a loss of memory or inability to form and maintain that memory. What I have shown you is that they do not have LTP. If I were to make that jump of memory, we need to actually do a behavioral task. The task is again a water maze experiment. Clearly you can see they can learn, their mean latency does come down. However, when you actually ask the question of how well they do in the probe trial, remember probe trial is the trial where we are actually taking away the platform, alright, taking away the platform and asking where does the mice spend most of its time searching. Okay. So, the legends for this different graphs are given down. So, this dark is the training quadrant right above here, and rest of them are the quadrants surrounding that, and you can actually see the wild type mice, that is the mice with everything being normal, no gene deletion, they search significantly more in the target quadrant, while compared to the rest of the place, while the mutant mice do not do that at all, they search equally well, when they do not distinguish the target quadrant. In fact, it becomes even more striking when you make a slightly different measure. Here now you are looking at, you are saying the you are actually measuring platform crossing. This is a measure where you are saying, hey look, if hypothetically if the platform were to be present in this, in the other quadrants, right, you can draw out the platform in those regions and then ask how many times that place has been crossed by this mice. Okay. That is another measure, right, because if the animal were to remember really where the platform is present, then you would expect that such a crossing to be higher in the target quadrant compared to the rest of the quadrant. And in fact, that is what you see in the wild type, no doubt. The black bar is the tallest among the rest of them. So, that is clearly higher, but when it comes to the mutant, they are not making that distinction at all. They are doing, they are moving around that place almost as if as the other how much ever they are moving in the other quadrants, clearly demonstrating that they do not have memory, they do not have the ability to form the memory. That is known to be dependent on hippocampus. This autonomous memory is known to be dependent on hippocampus. So, with this I have taken you through a reasonably long journey of starting from HM, where we said memories are formed in hippocampus, a particular kind of memory are formed in hippocampus, and if you remove that region, like completely taking away that region, what happens to those memories and what happens to the HM's ability to form those memories. From there to a place now we go ahead and manipulate a gene and remove a gene and show that the animal's ability to form these kind of memory is affected. Now, it is time to look back and ask what else can we learn from HM. If you are wondering, what what aspects of the ChemK2, we talked about the aspects of ChemK2 that really really can identify as memory molecule, but what other aspects of ChemK2 can be serving the neuronal function. There are several hypotheses that has been put forward, ranging from insertion, stabilization of the AMPA receptors that are present already in the postsynaptic neuron, to activating more AMPA receptors, to inserting more AMPA receptors, and so on and so forth. But there is one very important character that is going to prove it to be extremely crucial, that is differential distribution of ChemK2, alpha ChemK2 across the brain. Hippocampus seem to be having a higher concentration of this alpha ChemK2 compared to cortical regions. Now, keep this in mind. We will come back to this very fact while we are revisiting HM. Let us look back at HM. We remember this picture. This is the picture of HM, Henry Monition, who went through neurosurgery, brain surgery, to cure his epilepsy, where we have removed, people have removed the medial temporal lobe. From then on, he lost his memory, he lost his ability to form new memories, not of all kinds of memory, but a specific kind of memory. Thereby we came up with this wonderful classification of memory scheme, and thereon we went on to relate it to how we learn and so on and so forth. But there is one aspect that we did not touch upon. What we did not touch upon is the fact how he was able to retrieve his old memories, the memories that required hippocampus for its initial formation, but now the hippocampus has been removed, and still he is able to retrieve. Say for example, he is able to retrieve what his name is, what his family is, where is his home, so on and so forth. What could be happening here? That introduced the notion of a process called systems consolidation, wherein the hypothesis is that initially the information coming from various different cortical modules are getting integrated in the hippocampus and form a integrated picture of an event or a memory in the hippocampus, but over a period of time the memory that is formed in the hippocampus is such that it is able to assist or help the cortical modules to form an independent reasonably independent trace of memory for this event, such that now when you go ahead and remove this hippocampus much later, you have these memories that are formed in the cortical region that help you to retrieve that retrieve this event. But remember, to form this you need the hippocampal memory, and that is where the hippocampus is really required. If you remove the hippocampus right here, there is no integrator, there is no formation of that episode or an event in your life. So, thereby there is no memory at all. However, over a period of time, now you can actually go ahead and remove this, because this is independent of that, you can retrieve that independent of that. Now, that is an hypothesis. How would you test it? Now, you could test it, we already have some evidence that HM is actually able to retrieve this memory, but the point is you want to have this model outside of this patient, right. That is really a proof of the idea that you can this is what is happening. Once you have this model, then you can start to probe various characteristics of this model, of this hypothesis. And the model per se came from again in 1992 from Michael Fanslow's lab at UCLA. what they did was that they made use of the fear conditioning paradigm. Specifically, they made use of contextual fear conditioning paradigm, wherein if you take these animals, rodents, and then put them inside a box, we have seen them before, and present them in food shock. Now, there is no apparent CS, you are not presenting any stimulus externally apart from placing the animal in the context or in the box. This, in the absence of such, we have seen already that what we used to call it as a background, or in this specific case what we can call it as a context, the setting of this box, the lighting, the various olfactory and light cues and all together form an integral representation of the context here, which is the CS. The animal makes an association with this context to the shock. Now, what will be the result? Later on you bring this animal back to this context and you test it, the animal expresses fear, and you can score that fear, expression of the fear, by measuring freezing and then ask how well does the animal remember this. Very good. Now, what they showed is that such a remembrance or such retrieval is dependent on hippocampus, but that dependence is very short lived. So, what you could do is that when you are actually so what you are seeing is that you are measuring the freezing as a function of number of days after which you are performing a hippocampal lesion. Okay. So, there are two different controls here. One is cortical lesion, I mean just to remove a similar amount of volume, just to show that it is nothing to do with the act of lesioning and handling the animal through the surgery and all that stuff, removing the equivalent aid. This is called the double dissociation experiment, where you do that lesion in some other region and then you really show that that really does not affect it. However, if you do the hippocampus lesion right there in about a day, you do not see the memory, clearly showing this data clearly shows that the contextual fear expression is dependent on hippocampus. If you remove the hippocampus, there is no expression of contextual fear. So, that later on they have gone to show that what is stored in hippocampus, particularly the dorsal hippocampus here, is really the representation of the context. So, that context memory is gone, but it is gone only if you do this lesion in about a day. If you were to wait for long enough period of time, say about 14 or 28 days, and then do the lesion, the hippocampal lesion guys are as good as the control. They both can express fear for the given context, for the training context, almost equally well. Here is our model of a HM. This is the point at which HM has lost his hippocampus, but is able to retrieve his old memories, because in mice it is 28 days old. This is the point in HM where he is not able to form any new memories, because you are lesioning, or the whole training paradigm was 1 day old, it is new. So, through very simple but yet a powerful demonstration of lesion and the behavior, they have established a wonderful model that mimics this process of systems consolidation in rodent animal, paralleling HM. This started a sprout in memory research of particular kind. A memory, it is called as remote memory. These are memories, we call it remote memory because these are memories of the events that happened remotely, that happened some time ago. What kind of research are they? First thing you want to ask is, yes, it is good that it can actually retrieve this memory, but you want to ask how good are this memory compared to their original memory. Are there exact same details or is there something different? Now, if it were to be the fact that you are there is a different learning system and there is a different substrate than the original substrate, what is the likelihood that the different new substrate that is representing this memory, older memories, is going to be same as that of the original memory? If it is so, why even have that kind of duplication? Brian Wilgen in Alcino's Silva's lab asked that question of how similar these memories are. In order to do that task, what he did is he took these animals, all right. We know that if you lesion out the hippocampus, they show the time dependent consolidation effect, but then he asked a slightly different question. What I am going to do is that I am going to put these animals in a slightly different context than the training context. Okay. So, the slightly different is the key word here. How slightly is slightly? It is slightly that when you actually look at this put these animals and ask the question, do you remember this context or this context looks familiar to that, then the response of that animal would be it depends. It depends on what? It depends on when you ask the question. So, if you had to ask that question, that is you put this animal in a training in a novel context that is similar to a training context, if you had to put that animal right after you have trained them within a day's gap, they can clearly tell the difference between which is the training context and which is the novel context. You see this difference. However, that difference diminishes away as you proceed in time. Similar time scales as that of the system consolidation, you see at about 30-day time point, when you ask this animal, can you say if it is a training context or the novel context, they say I do not know. It is not that they cannot differentiate the two contexts. See, that is different from telling apart in which context they got a shock. See, these two are different. I want to make that distinction clear. It is one thing to say A is different from B, and it is other thing to say I am not sure it is the A or the B which is relevant. Is the second type of memory that memory loss or memory deficit that these animals are showing. How do we know that? We can actually put them in a different context, a context C, which is dissimilar to A and B. You will see they are at baseline, and in fact what you can actually also do are a few other cool experiments. You can give a small remainder somewhere in between, and then they will definitely tell the difference between A and B. You can also do few other characteristics which tells if they can separately separate the A and B well. That seem to be unaffected at all. What seem to be affected is their ability to say in which context they got the shock, not to say which context it is. This put together with the fact that you see the hippocampal dependence vanishing away in similar time scale. Let us put these two graphs together simultaneously, one after the other. If you put these two graphs right next to each other, what you see is that similar time scale and similar characteristic, suggesting that the memories that you retrieve after hippocampal lesion from the cortex may be having this property. That is to say, they have, the animals seem to have generalized the CS on which they got trained. It is called the stimulus generalization, that they are they have generalized, they have transferred the fear to a novel context. The ability to discriminate in which context they have got trained is lost. Now, why is that happening? Is that they lost the details themselves or something else is happening? It turns out you can do a different experiment here. What you can do is you can do a discrimination training. So, right from the beginning you train them in A as a shock context, alright. Remember our discrimination training is the CS plus and CS minus training. So, if you were to say A is CS plus and CS minus is B as a function of time. So, in the recent time you actually do this. So, they learn that for B they freeze less, for A they freeze more, and then you come back after 30 days and then ask how well they remember. They clearly remember A is the shock context and B is a novel context or is a no shock context. So, there is no problem in their ability to discriminate A and B. If you teach them to discriminate, they will discriminate, they will remember this details very beautifully, no problem at all. But left to themselves, the details seem to be getting muddled. To explain this, one of the proposition was from Franklin Lab that if you are having multiple overlapping memories or multiple exposures to a same event, then what the system consolidation process might be doing is they may be picking out commonalities between all these events as against those things that are different. If they are picking out the commonalities, then that is one way why you may be generalizing and not discriminating at a remote time point. Now, how do you test it? First thing is to prove that they are capable of picking out these commonalities. So, they went back, we go back to the water maze experiment, and now they said, hey look, I am going to make a small modification to the water maze experiment. Now, I am not going to place the platform in one particular location. Okay. What I am going to do is that I am going to move this platform around a particular probability distribution, that is one time I will be here, I mean so 10 times I will be here, one time here, one time here, like that. So, these are the exact locations where the platforms are placed. So, you can clearly see dominant amount of platforms were placed in this quadrant, a small amount were also present here, and then you train them. Mice, it is inordinately difficult, but the mice learn that okay, today this is where the platform is. How do we know that? You come back a day later. So, they have initially trained them in this set of platforms, and then they got into this introduced this platform in a new quadrant, right, these 3 places. Now, they come back after a day and then ask where are the mice searching. You can clearly see they are searching in the quadrant that they have been trained on lately, that is recently, right, one day. This is the last training that they have received, and that is where they are actually searching. However, you wait for about a month or so, and then now you ask where are they actually searching. What you see is that they are predominantly searching in the quadrant where there is a highest probability of finding the platform. It is not about which quadrant, but it is about the probability in which quadrant do I have a highest probability of locating the platform. Clearly giving in the clues that they are capable of extracting the commonalities given enough time, and that is probably what is happening when you are giving enough time and then when you are forming these representations over time across time. So, we address the question of what happens to this memory over a period of time. Now, we can step forward and ask, is there a molecular correlate here too? This is where the statement that I made right at the end of the alpha ChemK2 study becomes important. I told one of the beautiful characteristics of this alpha chemkinase is that a differential distribution of these molecules across the brain. these are hippocampus is highly enriched in the alpha MK2 compared to the cortex. So, given this, they said alright. Now, when we knock out a gene in an animal, we know that the genes come in 2 copies. So, clearly you can take away one of the genes and leave the rest, leave the other, or you can not take away anything, or you can take away both of them. Depending on that, you will call them as heterozygous, wild type and homozygous mutants. The alpha MK2 that we looked at were all homozygous mutants, that is, the alpha MK2 has been deleted completely, both the copies of the genes have been removed. Now, if there is a differential distribution of this concentration, now if you go back to the heterozygous, then we can see we have an ability to specifically look at the effects, the concentration effects much more in a much more pronounced manner. So, Paul asked this question and then said, hey, look, we know cortex takes part in remote memory formation, and we have a way of modulating this differential response through alpha MK2 heterozygous mutants. Since cortex has less anyway, now if you remove even half of it, that can bring down the alpha MK2 concentration below the levels that are required for plasticity formation. So, they may not be good enough, that the levels may not be good enough to form remote memory, while on the other hand in hippocampus anyway it is enriched in alpha MK2. If you reduce by half, maybe you get away with that. Now, how do you test it? You take these heterozygous mutants and then run through the behavior, right. And what do we expect? We expect hippocampus dependent memory, that is the recent memory, to be intact, because if things were to be normal and that is half concentration is sufficient to see through, you should be able to see through, and in fact we know that half may be sufficient enough, because the remote memories do form in the cortex, while at the cortex that may fall below the critical level, making the animal not be able to form the remote memories. This is inverse of the HM problem, right. The animal would be able to form the recent memory, but will forget, will not be able to keep it for longer period of time. Okay. What do we see? So, they took these mice, so and then subjected to the water maze experiment. When you do that, you can actually see these are the comparisons we are talking about, wild type versus the alpha MK2 heterozygous mutant. The plus slash minus represents that. If it is totally minus minus, it is a homozygous, plus plus it is a wild type, plus means the gene is being present, minus means it is not present. So, when you are looking at the heterozygous mutant, what you see is at the end of the training, at the end of 3 days, they are beautifully learning, no problem at all, they are specifically searching in the target quadrant, pretty much like the wild type. However, when you bring them back after 17 days, you do not have to even wait for a month, you bring them back after 17 days, what you see is that compared to this, not just the water maze, you can actually do this experiment on fear conditioning too, and that is what you see. In the recent time point, they remember very well, no problem at all. However, if you wait long enough time, or in fact if you wait for reasonable time, any reasonable time, right after 10 days, they fail to retrieve these memories. Here we have created a new kind of memory deficit, wherein the recent memory, hippocampal dependent recent memory is absolutely fine. However, the ability of the animal to retrieve this over a long period of time, the remote memory, is lost. With that, I would like to end this lecture now, and then we will see you in the next lecture and I will describe to you there what are all the recent advances and how and where we have reached to the point where we can actually start to manipulate this memory neurons participating in memory. How do we do that, and what effects do you see when we do that will be the part of the next lecture. Thank you.